Hi, this is Rethik, your biology subject matter expert from the MemoNeet, and once again, welcome to the biology series. So, in this video, we will be studying on the regulation of gene express and lack of lactoperon. What is it, and what is the major role or function? That is what we are going to study. So, basically, we have two types of organisms. One is eukaryotes and prokaryotes. So, in both, we will be seeing something different way of gene expression, right? So, in eukaryote, you will be seeing like transcriptional level, that is primary transcript formation, then processing level, that is slicing, regulation of slicing. Then we have transport of mRNA from the nucleus to the cytoplasm, because inside the nucleus you will be seeing the genetic material, and there this mRNA, which is nothing but the messenger RNA, is produced. So that need to transport from the nucleus to the cytoplasm, right? So if this is a cell and you have a nucleus, here you will be having the genetic material, say like it is cytoplasm. So from the nucleus, the mRNA should go to the cytoplasm, then the translation level. See, as we know that in genes in a cell, right, they express or they come up performing a specific function or a set of function. So every gene in the cell, they perform its own specific function. For example, if an enzyme is called as beta galactosidase is something an enzyme which is actually synthesized by E. coli, right? So which is actually synthesized within or by E. coli bacteria. So it is used to catalyze the hydrolysis of discharge, that is lactose into galactose. So that is nothing but it acts as what? Catalysis. It acts as a catalysis where the hydro catalyze that is used to catalyze the hydrolysis of, that means from lactose to galactose, and then later on to the glucose. So what happens during this process? The bacteria uses them as a source of energy. Okay, the bacteria is going to use these enzymes as a source of energy. Hence, what happens if the bacteria do not have this specific thing called as lactose around them, then they will not be able to utilize the source of energy, right? So if around them, if they do not have lactose around them to utilize the energy source, they would no longer require the synthesis of enzyme, that is beta galactosidase. So if there is a presence of lactose, then what happens? The lactose should get converted into galactose, then later on to the glucose as an end product. So for that, it requires an energy, and this specific enzymes, that is beta galactosidase, is an enzyme which is been utilized by this E. coli, like during the process, or it acts as a catalysis. That is, it gives a source of energy to the bacteria for the conversion from the lactose to the galactose, and later the glucose. So if lactose itself is not there, then it doesn't require the energy. Like if lactose is not there, there is no subject or there is no matter of conversion. So conversion is not there. That means it doesn't require energy. So therefore, if energy is no longer needed, obviously the synthesis of beta galactosidase, that is the enzyme, right? It is no more needed or necessary. So the synthesis will not happen. Therefore, in a very simple time, if I need to explain you, it is a metabolic, physiological, or environmental condition that regulates the expression of what? Gene. So the development and differentiation of embryo into adult organisms are also a result of coordination, regulation, or expression of several sets of genes. So in what way the genes are expressed, based on that itself, the embryo development from the embryo, initial embryo, to the adult takes place. Now, whereas in prokaryotes, what happens in prokaryotes? So control of rate of transcriptional initiation is the predominant side for control of the gene expression. See what happens in a transcriptional unit, the activity of RNA polymerase. Okay, so the specific RNA polymerase, which what is the major function of the RNA polymerase? It acts as a promoter here. So RNA polymerase, what it gives, it at a given promoter, what happens with the promoter? So obviously, if there is a single stranded DNA, what happens? There will be a promoter region will be there. So RNA polymerase will come and bind to the specific site. So in presence of promoter, what happens? So that it is in turn regulated by so obviously the synthesis of the strands will takes place. So how this all thing has been regulated? They're regulated by the interaction with the accessory proteins. So what are these accessory proteins? Like which actually going to this accessory proteins, which affects its ability to recognize the starting site. That is what RNA polymerase will go and recognize the starting site, codes with it in presence of promoter. So what the promoters will, push the RNA polymerase. Chalo, do it. Okay, start, synthesizing the strand now. So in that way, it is going, the promoter acts as a so it promotes the synthesis of the another strand, that is complementary strand. So this regulatory protein, can act as both positive and negative. So positive is nothing but activators and negative is nothing but repressors. Okay. So what is this? See, if there is a promoter region, so that is accessibility of the promoter region in the prokaryotic DNA, is in many cases it is going to regulate it, so that has been regulated by the interaction of the protein with sequences. So we normally term them as what? Operators. So the operator is actually a region. We need to understand the terms here so that in coming lac operon you'll understand it even very easily and simply, simple formed. Okay, so the what happens here, that is that is operator as a region which is adjusted to the promoter element in most operons. Okay, and as most cases the sequence of the operator binds or repressor proteins. So they're going to bind or repressor protein, and each operon has its own specific operator and specific repressor. For example, if we are taking a lac operon as an example, what happens here? We have a lac operon. Okay, so lac operon. So there is a presence only in the lac operon and interacts specifically with the lac repressor only. That means, so there will be one is activers will also be there. Activers is a kind of a protein which regulates the overall reaction. So there is repressor, that is negatively. So repressor will be there. So that will specifically go and bind to that protein itself. So it will stop the, sorry, specifically go and bind to the region, and it is going to stop the entire process. Say like if you have taken the lac operon as an example. So in lac operon, we have what? Lac repressor will be there. So that is what initially it is going to interfere with the process and all. So going or moving to the slack operon. So that was actually illustrated or described by, as a result of close association between the genetics. So we have some scientists, Francisco and, they Jacoba and some of the biochemists. So they have, explained this lac operon model. So they were the first to describe this transcriptionally regulated system. What happens in lac operon? So here the lac is nothing but it refers to the lactose. Okay, so when I told you like lactose, what exactly comes here? Polycystronic structural gene is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as what? Operon. So to name for few example, we have lac operon or TRP operon, ARA operon, HIS operon, VAL operons, and many are examples. So we'll come with lac operon. What is this lac operon? How the genes are, they have been regulating the transcription process and all. See, in absence of inducer, so when there is no inducer, what happens? So that is what we are going to see now. See, the lac operon consists of one regular gene. I can call it as regular gene, that is I gene. Here, I does not refers to as inducer. We need to remember one thing, that I does not refers to as inducer. Rather, it is derived from the word that is inhibitor. So I can call this I as an inhibitor. Okay, and three structural gene will be also present, that is Z gene, Y gene, and A gene. Okay, so this is one thing. So here what happens, I gene codes for repressor. So this is what the repressor of the lac operon, right? So I gene actually codes for what? Repressor. That means of the lac operon, and the Z gene, So we have the Z gene. So it codes for beta galactosidase. So Z gene codes for beta galactosidase, then which primarily responsible for the hydrolysis of the discharge or lactose into monomeric units, then galactose and glucose, something like that. So that is what responsible, beta galactosidase. Then we have another one, that is Y gene. So Y gene codes for what? It codes for permease. Okay, so you need to understand I gene codes for receptor. Okay, or what you can say, I gene codes for repressor. Okay, so I gene, as you can see, it is coding for what? Repressor. Then Z gene, it codes for beta galactosidase. Then Y gene is present. Y gene codes for permease. Okay. So what is the presence of permease, which increases the permeability of this beta galactosidase in the cell? Okay. Then the gene encodes for the so this is what normally happens. So we have three genes here. Hence, all the three genes produced in lac-operon are required for the metabolism of what? For the metabolism of lactose. So here Z gene represents, so it codes for beta galactosidase, Y gene codes for permease, and A gene codes for trans acetylase. Okay, so this is the thing. So all these three gene, they are required for the metabolism of lactose. So what happens here? So lactose is actually a substrate for the enzymes beta galactosidase and is required to switch on and switch off of the operon. So inducer, where lactose is a substrate for the enzyme, okay, where this beta galactosidase we are getting, right, and enzyme beta galactosidase. And what it does, it is helping in regulating the switching on and switching off of the operon. So now this lactose, I can in lactose, okay, I can call it as what? Inducer, because it is the one which is actually helping in switching off and switching on. So in absence of carbon source, such as if there is no glucose, if lactose is provided in the growth medium, so in presence of lactose to the bacteria, what happens? The lactose that you are seeing, right, in presence I'm talking, okay, remember here. So what happens is lactose is transported into the cell through the action of permease. Okay, this lactose then induces the operon in the following manner. So what is it? We will see now. So the repressor in the operon is synthesized. So is this the in a normal condition? So repressor from I gene is being synthesized, right? From the I gene. So now this repressor is protein is going to bind to the operon region. That means under normal condition, what happens? uh, yeah. So under normal condition, what happens? These operon, okay. So obviously this repressor, right? It is going to bind in absence of inducer. I'm saying it is going to bind what? O region. So once it goes and bind to this O region, is nothing but the operon region. So what happens? So sorry, you will see that there is no function going on, right? So in presence of inducer, so if inducer is present, so what happens? So now inducers will come and it is going to bind this repressor and make the repressor inactive. So now the repressor is in inactive mode. Okay. So as a result, what happens? So now this will allow the RNA polymerase. So there will be RNA polymerase, right? So it is going to allow this RNA polymerase to access the promoter, then and transcription process will take place. So in transcription, now they are free because there is no inducer is there which is going to bind this operon region, O region, so that now origin is free. So there is a promoter here. So it is going to help in the transcription process, right? So as a result, I told you there are three genes now, Z, Y, and A. So now gene, it is going to synthesize, Okay, lag mRNA is nothing but messenger RNA. So Z gene is going to synthesize beta galactosidase. Y gene is going to synthesize permease. So permease, what is its major function? It will help the beta galactosidase permeability. It gives the permeability to it. And last, that is A gene is going to synthesize. That is translation process will happen and it is going to synthesize trans-acetylase. Okay, trans-acetylase. So one thing we need to remember here is lack of peron can be visualized or regulation of enzyme synthesized by its substrate. So glucose or galactose, which will be present, right, that cannot be act as an inducers for the lack of peron. This is very important thing. Then regulation of the lack of peron by repressor is referred as what? Negative regulation. That is regulation of the lack operon by repressor, okay, is referred to as negative regulation, and lack operon is under control of positive regulation as well as. Okay, so that means both. If it is inactive state where inducer is not there, so what happens? And if inducer is there, what happens? If inducer is there, obviously the transcription process will occur. If inducer is, not sorry, not there, then obviously transcription process will not occur because what happened? That repressor, protein that is going to come and bind to this um, operon region. So that is what we need to understand. This is switch on and switch off model. Okay. So switch on model, switch off model. The next topic we have is replication. What is this replication? See, we know the DNA structure, that is double helix structure, which was being proposed by Watson and Crick, right? So where they also proposed a scheme for replication of the DNA. So to quiet their original state, so it has not escaped our notice that the significant pairing we have postulated. So like whatever the genetic material copied, like how exactly one single strand will be there, exactly opposite, like same another strand will also be made. That is what they have explained. So that also suggests that two strands would separate and act as a template for synthesis of new complementary strand. That is nothing but if you have a DNA here, so this is one strand and this is another strand. So individual, each strand has the ability to become what? A separate strand and help in acts as a template strand and helps for the synthesis of new complementary strand. That is what they're saying. After completion of the replication, what happens? Each DNA molecule would have one parental strand, that is the original strand, and the another strand will be newly synthesized strand, the strand that is newly synthesized from the parental strand. So this scheme was termed as what? Semi-conservation DNA. Okay, replication. So whenever you a question arises from semi-conservation DNA, replication is nothing but semi-conservation is nothing but semi is nothing but half conservation. Half is conserved. So out of the DNA, the parental strand, that is the two strand gets separated, and each strand becomes what? A template strand, where another newly synthesized a strand will be synthesized from the parental strand now. So that means half of the DNA is conserved and another one half is newly synthesized DNA. So one experimental proof has been conducted to showcase how the semi-conservation thing works. Okay, we will try to see that thing. Right. So the experimental proof is yeah, Stahl and Me- Meselson, so they are the scientists who worked on this experiment in 1958. So they grew what? E. coli bacteria in a medium containing 15 NH4Cl. Okay. So here N is 15 N, that what we are saying, right? It is actually what? Nitrogen, which is heavy isotope of nitrogen, as only nitrogen source for many generation. So it is isotope means radioactive kind of thing, nitrogen. Then the resultant was that 15N is nothing but nitrogen was incorporated into newly synthesized DNA as well as other nitrogen containing compound. So here you have what? N nitrogen. So which is nothing but it is heavy isotope in nature. And another one thing, we have a normal one. Okay. So what they do is this heavy DNA molecule could be distinguished from the normal DNA by centrifugation. So they could be distinguished easily by using centrifugation, because 15N is nothing but the one which is heavy isotope nitrogen, and another one is also nitrogen, but it is normal DNA now. So how we can distinguish? By centrifugation method and observing them, right? So where you are center, obviously we will be using density gradient centrifugation, and it can be separated from this 14N DNA, that is natural DNA, based on their density, that is based on their density. So one thing that we get to know here is they took a transferred cell. So we have here, so obviously they were mixed. As a result, in first generation, you will be able to see a kind of, one is parental DNA and another one is newly synthesized DNA. The red one is parental DNA. The green one that you are watching is newly synthesized DNA. Okay, so then they transfer the cell into the medium with normal 14NH4Cl and took a sample of various defining time interval as the cell multiplies. And from those cell, they extracted the DNA, which remained as a double-stranded helix itself, and various samples were separated independently as the centrifugal gradient to measure the density of the DNA. After that, so the DNA that was extracted from each culture, each cell, right, or from one generation, so they were being transferred from like 15N to 14N media, that is nitrogen isotope, to the normal media. Okay. And they were left for like 20 minutes. And after 20 minutes, this E. coli divided 20 times than the normal original population. And some were been hybridized, or and some were been intermediate density was showing. So that is DNA extracted from the culture after another generation, like after 14 minutes. That is, I can call it as what? The second generation was composed of equal amount of this hybrid DNA and light DNA. So I can call them as hybrid DNA and this as light DNA. That means one thing was very much similar, that when, so here again this DNA, during replication, both that is daughter DNA or two double-stranded DNA got separated to become single-stranded DNA, and each DNA, started synthesizing its own new DNA. So the heavy DNA synthesized same one like it, or what you can say, and the one which was the lighter DNA synthesized exactly one like it. So very similar experiment involving using a radioactive thymidine to detect the newly synthesized DNA in the chromosome was performed on one of the way, that is Vicia faba, so which is a kind of a beans, by Taylor and his colleagues in 1958. And the experiment actually proved that the DNA in chromosomes are also replicate semi-conservatively. That is nothing but half of the DNA will be retained and another newly synthesized DNA can be absorbed. That is what they are trying to say in this experiment. So this was all about how the replication happens and the experimental proof regarding the replication. I hope this video is clear. This particular topic is clear in the molecular biology. So with regards to any doubt, you can contact me through the comment section, or you can also contact me through the Telegram channel. So link is already in the description. And yes, I'll be looking forward for your likes, comments, and also do subscribe to the channel and share with your friends. Okay. Thank you once again. Bye.